Hey there, science fans. Today we're diving into the incredible world of DNA and how it shapes who we are. DNA, or deoxyribonucleic acid, is like a blueprint for building and running our bodies. It's inside almost every cell, holding the instructions to make us unique. Imagine a massive library filled with instruction manuals. That's what DNA is like. These instructions are organized into genes, which tell our cells how to make proteins. Proteins are the workhorses of our bodies, doing everything from building tissues to fighting off infections. DNA has this cool, twisted ladder shape called a double helix. The sides of the ladder are made of sugar and phosphate molecules. The rungs are made of special chemicals called nitrogenous bases. There are 4 types of bases: adenine, thymine, guanine, and cytosine. These bases have a special pairing rule. Adenine always pairs with thymine, and guanine always pairs with cytosine. This pairing is crucial for DNA replication and protein synthesis. Like puzzle pieces fitting perfectly, this ensures that genetic information is passed on accurately. Think of it like a secret code that only DNA knows how to crack. So how do we go from DNA to protein? It's a 2-step process: transcription and translation. Think of DNA as a master chef's recipe book locked away in the restaurant's office. The chef can't cook from that. They need a copy they can use in the kitchen. That's where transcription comes in. Imagine a messenger carefully copying down a recipe from the master book. That's like RNA polymerase, a special enzyme, creating a messenger RNA, mRNA molecule from the DNA sequence. This mRNA is a single-stranded copy of a gene's DNA sequence. It carries the genetic information from the nucleus, the cell's control center, to the cytoplasm, where protein synthesis happens. Think of it like a courier delivering the recipe to the kitchen. Now, we need to translate this recipe into a delicious dish, or in our case, a protein. That's where translation comes in. The mRNA molecule acts as a template for protein synthesis. It's read in groups of 3 bases called codons. Each codon specifies a particular amino acid, the building blocks of proteins. Remember those bases? They're like letters, and codons are like 3-letter words that tell the cell which amino acid to add next. But how does the cell know which amino acid matches each codon? That's where transfer RNA, tRNA, comes in. These molecules are like delivery trucks, each carrying a specific amino acid to the protein assembly line. They have a special sequence called an anticodon that matches up with the codon on the mRNA. The protein assembly line is called a ribosome. It's a complex molecular machine that reads the mRNA codon by codon, matching each one with the correct tRNA and adding the corresponding amino acid to the growing protein chain. It's like a factory floor where all the parts come together. The ribosome moves along the mRNA, linking amino acids together like beads on a string. This process continues until the ribosome reaches a stop codon, signaling the end of the protein. The newly synthesized protein then folds into a specific 3-dimensional shape, ready to do its job. The genetic code is universal, meaning the same codons code for the same amino acids in almost all living organisms. It's also redundant, meaning multiple codons can code for the same amino acid. This redundancy provides some protection against mutations or changes in the DNA sequence. From the tiniest bacteria to the largest whales, all living things share this incredible system of genetic information flow. It's a testament to the power and elegance of evolution. And it underscores the interconnectedness of all life on Earth. So there you have it, the amazing journey from DNA to protein. It's a complex process, but understanding it helps us appreciate the intricate workings of our cells and the remarkable molecular dance that keeps us alive and kicking. Stay curious, science fans!